In this presentation, Dr. Stephen White, research geneticist with ARS since 2006 and adjunct faculty member in the Department of Veterinary Microbiology and Pathology at Washington State University, discusses the identification of genetic markers in goats that show resistance to Scrapie and what this means for the goat industry. The American Goat Federation, AGF, was organized as a non-profit national association in 2010 to represent the interests of all organizations and producers engaged in the production and marketing of goat milk, meat, fiber, pack goat, and grazing services across the United States.
Thank you very much for the kind introduction and the invitation. I'm excited to get to present what we think is good news on a long-running experiment with goat Scrapie susceptibility and resistance in goats. So, a little bit of quick background. Obviously, most of you in the room know most of the background on Scrapie, and Diane's done a great job setting this up. But the issues with Scrapie, so it's been around for a long time. It's been known since the 1700s. The transmissible part of this is actually the proteinaceous infectious agent. This is a protein that's made in the nervous system of every mammal that's ever been sequenced or studied. And what you have is two forms of the protein. One of them is what we call the PRP cellular or the PRPC. It's the native conformation. It's the way the body actually makes the protein to fold and to work. And then the refolded form is the PRP Scrapie. It has two properties, one of which is that it's extremely resistant to degradation. That means that in the animal, the body's own natural systems can't go take it apart the way they would normally with any other protein. And then obviously, so that leads to the pathology and even its persistence can contribute to the transmission. So in terms of diagnosis, we're looking at the brain or the lymph nodes are basically our options. In the brain, after death is a really good way to go and try to diagnose and to see what's going on. But the brain's not really a live animal test. The animal's already got to be gone by the point that you're actually getting to that good sampling. And so what you can do is you can do lymphoid tissues. And so these are rectal biopsies, for example. Some in the old days we used to use a third eyelid test occasionally, especially in sheep. And one of the issues we have is that the lymphoid involvement, so whether or not the diagnostic test is going to work, is a little more variable in goats, at least mostly most of the data that we've seen. So background again in this audience, the variability, I guess, is the take home from this slide. The variability is different in goats than it is in sheep. So in sheep, it's pretty well defined most of the time. There's some odd genotype issues that create some exceptions, but not super often. The incubation time is relatively straightforward, and we're starting to understand why in goats sometimes the incubation time can be more variable. There's a couple genotypes that can extend incubation. They're relatively common. They're still not common, but they do impact things. The lymphoid tissue involvement is an issue, and then also placenta transmission. And so when we started this experiment we really started 10 years ago in 2007. And that was when we were first acquiring animals and trying to set things up. At the time that we started, there was no strong resistance, which obviously has led to difficulties in terms of management with the goats. So everyone in this room, from the discussion I've been hearing, you're very familiar with this story. And so the ARR haplotype confers the strong resistance to classical Scrapie in sheep. The VRQ is highly susceptible and changes incubation time, odds of infection, and so forth. And so, as Diane was saying, the eradication, you've got genetic tools, at least in sheep, but not in goats. And so clearly the disease issue in goats. It still happens. It's still happening even in the United States. And so in terms of the needs, what do we need to do? Well, obviously it'd be great if we had a completely resistant genotype. And then we also need to know about incubation time, because if it's really, really long, we need to know that too. So, in terms of where we are, we did a national survey looking at 10 different goat breeds. And we were looking, based on when we first started this, we were starting looking at the natural Scrapie cases, comparing them to their genotypes, and looking for genotypes that were underreported or actually non-existent. And what we found, looking around the world, there were 2 different alleles, and we found very big differences in allele frequency. The 2 alleles, one of them is at codon 146. There's a serine, so this is an S146 animal. And so what we found is that this is really common in the US. This is, out of the 10 breeds that we surveyed, we found the allele present in 7 of them. It probably is present even in some of the other ones. We just didn't sample enough animals. And even within the breeds, it's really common. So this is 1 out of 8 of all the alleles we've sampled from the US. And so the likelihood is many operations will have this allele, and they'll know someone who does. So we've even seen some operations where a lot of the animals will even have 2 copies of it. The situation is a mirror image in Europe. And so, and by that I mean continental mainland Europe. It looks like there was a population bottleneck taking goats to the UK from continental Europe, and the US tends to track after the UK. And so in Europe, this allele is so rare that they can't even do statistical comparisons of the S146. They found it in Cyprus and some other places, but they happen to lump the genotypes together. It's really rare. So what the other thing we found is the mirror image of this. And so this is a lysine, so which is abbreviated K, so K22 at position 222. And this one is common in Europe. There's a lot of animals there, and there's a lot of interest in pursuing this as a potential resistance allele. And it's really rare, at least in the samples that we've seen in the US. And so we had only found this one in dairy goats, and we had only found this in a handful of dairy goats in the United States. We're talking 0.5% allele frequency, and so really rare. And so, but what we decided when at the outset was we did at least find both of these alleles in the US. There was potential to apply either of them. We weren't sure if either was going to be better or worse than the other. Let's try both. And so we decided to get started with an experiment using either the K222 or the S146 in separate animals. Along the way, I'll insert here that we got some good news after we got started. And so the animal experiments, as you'll see, we've been working for nearly 10 years on this. They take a long time, but some cell-free experiments that measure the conversion from the natural PRPC version of the prion protein to the PRP Scrapie form were done to try to look at the conversion efficiency. One of the things that was found with the sheep is that the ARR haplotype results in a protein that it's really, really hard to even try to force it to refold into the Scrapie form, which is probably why they're so resistant. And so what was done is a cell-free experiment is done elsewhere, Iden et al., and they found that each of these 2 alleles, both the S146 and the K222 alleles separately, were approximately on the same order of resistance in terms of the misfolding as the ARR in sheep. And so that encouraged us and strengthened some of the rationale for moving forward with these. So along the way, we've mentioned the different ways to do diagnostics, and so we're going to use live animal tests when the animals are alive. These are the lymphoid involvement, so the lymphoid tissue or rectal biopsy, and so forth. We're going to pay really close attention and look at animals. We've got Scrapie's been studied at ADRU for a long time, and we've got supervising veterinarians and trained staff and so forth looking for anything that resembles clinical signs. And then when we get to the point where an animal is put down either for Scrapie or for some other reason, then we're going to go and do brain tests as well. So, what was our design? We're trying to test these alleles. What are we going to do? The first piece, a lot of times in Scrapie research, people will do an intracerebral challenge. That is, they're going to put Scrapie straight into the brain. What we wanted to do was give a uniform challenge, but we wanted to do it orally because it's possible that in terms of actual on-the-ground situations that putting it straight into the brain bypasses an important protective mechanism that taking it in through the mouth would capture. And so what we did was we did it within 24 hours of birth. And the reason for that, with the protein infectious agent, in ruminants, the gut is very permeable to protein for the first 24 to 48 hours, and that's about absorbing the antibodies from mom. And so we wanted to take advantage of a high challenge situation when the kids would absorb Scrapie very quickly and easily, but we could still take advantage of some of the natural mechanisms. So what we did is we took a homogenate, so we wanted to make the dose as close to exactly the same as humanly possible, and we did this from a goat Scrapie case and from a placenta that had lots and lots of Scrapie. And so all this was goat source material. It was one goat, it was the same goat. So then we went and did some controls. So I mentioned our national survey of all the goats, and what we found was one haplotype in goats is by far the most common in the US. And so we called that haplotype 1. And so what we did was, of the controls we did, we made sure that they had 2 copies, they're homozygous, for haplotype 1. So these are your standard, straightforward prion genotype goats. And then we used the same group of controls and compared them to 2 experimental groups. The one group was the S146 group. These animals have only 1 copy of S146. They're heterozygous. And the other chromosome, they have haplotype 1, just like the controls. And so what we're measuring is the S146 1 copy versus no copies. And then in a separate group, we looked at the K222s. And in these, again, we're looking at heterozygotes. They've got 1 copy, just 1 of the K222, and 1 copy of the common haplotype 1, just like the controls. And so it's 1 copy again versus none. What did we find? Well, if we did our 5 homozygotes and our controls, 4 out of the 5 became rectal biopsy positive. It wasn't too surprising that we had 1 that didn't go that route, again, because of the variability. All 5 of them progressed to clinical disease, and they lived an average of about 2 years. And so we literally only had one of these controls that made it past 24 months before it went clinical, and it went down at 27 months. And so we're really looking at 2 years. What this tells us, as far as the experiment, was that our inoculum that we used to challenge these animals was both infectious and consistent. So now let's think about, okay, well, what's been going on with the experimental groups? So the S146, again, 1 copy in each of these heterozygotes, 8 of them. As of today, none of them have ever turned rectal biopsy positive. And this is starting to be a really long experiment. Furthermore, clinical disease. There's been no clinical disease at all in these S146 animals that have one copy. The average time without any clinical disease is approaching 7 years on average, and they're still going. Literally, we ended up having to put 1 animal down last week. This animal was 7.5 years old, and it's negative. And so the comparison statistically to the controls is very significant. And as of today, 5 out of the 8 of these experimental goats are still alive, and as far as we can tell, Scrapie free. So this is encouraging. They're starting to be older goats, but from a Scrapie perspective, they're happy goats. There's no problems that we've been able to find, and we've tried really hard to, and given them even a demonstrably lethal dose of Scrapie. And so that's a good outcome for where we are so far in a long-going experiment. How did things go with the K222s? So again, we had 8, each had 1 copy that we started with. As of today, none of them have been rectal biopsy positive. Furthermore, none of them have developed clinical disease. In this case, there's not quite as many of them still alive. There's only 3 of them left, but a lot of these animals, all the ones that have gone down for other reasons. And so because some of them needed to be put down for humane reasons early from other diseases, our average isn't quite as far along, but it's still at this point actually is probably a few months older than that. So we're approaching 7 years on the Ks as well. And so again, from a Scrapie perspective, you've got happy goats. Some of them have had other health issues that have gotten old, but for Scrapie, they've been a really good outcome, really happy animals. So where are we in terms of age? So this experiment is still ongoing, but we thought it was important to start to pushing again to advertise, hey, this experiment's still going on. It's our understanding, and people in this room, you know better than I, but you correct me if I'm wrong, but a lot of the producers I've talked about have said that usually in terms of commercial operations, that culling by about 6 is pretty standard. And some people have data for that and other reasons. And so at least for a lot of the commercial animals, the disease-free periods for each of these genotypes are already starting to exceed productive lifespans. It doesn't mean that some goats don't get old. Obviously they do. We've had certainly individual reports of even teenagers. But in terms of on average, most of the time for a commercial operation, these animals are starting to really be pretty old. And in terms of trying to avoid a clinical Scrapie in the lifespan of the animal, we're starting to be there. So other questions. So along the way, this is a very long-running experiment. Very small number of animals that had either one copy of either of these alleles have actually been reported with natural Scrapie. Literally, we're talking about 2 of one genotype and 3 of another. In some of those cases, the other allele, so the other chromosome, had something wonky going on, and so did that contribute, did it not? We don't have much statistical ability to tease that out, but it's looking like it's not going to be an absolutely perfect resistance, but the question that we've had is in terms of the ARR in sheep. There actually are a handful of positive animals that had one copy of ARR that did develop classical Scrapie. So the question that we have are in these cases, they're rare, are they as rare as the ARR one copy cases? And we don't know. The data just aren't there yet in terms of the number of Scrapie cases to actually make a fair comparison. But it's we think, a worthwhile question. Another question, all of this experimental data, and actually there's a very recent report just in the last few months of an ongoing kind of natural experiment in Europe that they actually found some animals that had the S146 and they've been on premises that have had ongoing Scrapie for a while, and all of them are fine. So the next question is what about the 2 copy cases? To our knowledge, there's never been for either of these alleles a Scrapie positive among a 2 copy animal ever identified. And so if the 1 copy case isn't full enough resistance, what about the 2 copy case? And at this point, I think that's a very fair and open question. In Europe, where the KT22 is more common, there's been a lot of interest in pursuing, hey, look, KT22, 2 copies, is that the genetic ticket to make sure you never ever have a Scrapie problem? And the same could be explored in the US and UK for the S146. And the other question that we have is, how long is this going to keep going? If the resistance hadn't been as strong, then we would already have finished the experiment and published our final report, and everybody would know exactly how all of this went. The good news in terms of the resistance being identified is that the experiment is still ongoing. And so what we have are, this is another in-process update with our averages are approaching 7 years. And then we're still trying to go forward because at this point we can't say they're never going to develop it in their entire life. If they do develop it, the incubation time is going to be really long. That's what we know today. So we're encouraged. The data continue to get stronger with each passing month and year that this goes on, and we've actually started getting some interest from commercial companies in offering a genotyping test on the basis of these alleles. I talked to the company last week, and they were in process testing, but as of today the test is not yet available, but they're trying to go and make it available soon. If you have questions, please come talk to me, and we'll talk more about that, or even in the question and answer session. So in terms of thank yous, obviously there's a lot of people. I'm here talking, but a lot of people contributed to this. Some of you will know Catherine O'Rourke, who has retired, but she was a longtime member of ADRU. David Schneider, Jim Reynolds did a lot of the inoculations. Dan Waldron from San Angelo has contributed to multiple papers with this. Dr. Sinar is working on a new report to try to get the more updated data out so that everybody can see and use. Some of you will know Joni Rao, other members of the ARS and farm crews trying to keep all of this going. The good news, at least in this case, is that they've been maintaining healthy animals for the last several years. But now they're getting older, and so we're having to try to keep things going, and it's a little bit more intensive than it once was. But we thank you so much to all of them. Thank you again to the organizers, and thank you for your attention. If you've got questions, I'd be happy to entertain them. The question is that we've worked with these two alleles, and are there others that show promise? The answer is yes, sort of. Depends on what it is that you're after. There's an S allele at position 127, and it has extended incubation, but it only approximately doubles the incubation. It doesn't take it as far as these genotypes have. There's an M142 that does about the same thing. Are there really promising other ones? Yes, at position 146, so we're looking at the S146. There in Europe, in Cyprus, there's a D146, and it's also never been found, to my knowledge, in a Scrapie positive on Earth, but it's pretty rare in Europe, and we've never been able to get our hands on an animal that had it. And so there's a few more alleles out there that are they're either relatively common and their extension of incubation is going to be intermediate, or they're pretty rare but they could be very promising. And so we tried to go for the things that we could find and that people might actually be able to use in a breeding program, at least as a first step. Question is about making the 2-copy animals. Yes, and we had questions. Yes, no, and we had questions. So have we made homozygotes? Yes. And so some of these animals, especially the K animals, had to be pretty inbred just to go and try to get that. We did actually inoculate one, and it went for some years before being put down. It was negative. But in terms of going and looking long term, Another question that we have that's seen in some other TSE systems is the genotypes of the animal that you use for your inoculum versus the genotypes of the animal that's receiving, so the one that you're actually testing. And in some systems, the animals that have at least a partial genotype match will accelerate the TSE's the disease course. So for example, in our mind, the ideal case for an SS homozygote would actually be to get one of those possibly very rare S heterozygotes that actually had Scrapie and use its material as the inoculum. Because as we can see with this experiment, this has gone on for a really long time, even with setting it up in the beginning. That's why we did the one copy experiment, was to try to get some of the match going on and accelerate things if we can. And so similarly with the K. So what we had originally set out to do was to wait for these animals to become positive and then develop the 2-copy test. And so what's happened is, as we've gone on, they've never been positive. And so we've kind of still been on hold with anything more advanced on that score, but it's a great question. Yes, sir.
What government agency do you work for?
I work for USDA's Agricultural Research Service, so we're the in-house research arm for USDA.
So are you part of the original budget that I questioned before, or are you an additional?
We're separate funding within. So USDA, the overall department, we're within the research side for ARS. It's a totally separate budget. Thank you.
My question about looking at the goats, I know looking at the pictures, I think you used dairy breeds in this, or did you also look at meat breeds?
Yes. Good question. So the question is about the goats that we used and what their breeds were. Let me see if I can get back. So we did 3 different breed backgrounds, and so you can see. So there's Toggenburgs in the back. As it happened, the Toggenburgs were one of the breeds we found the KT22 in. And we found very few animals with KT22, and so we did use some dairy animals. But actually, in this experiment, the majority of these animals were boar cross animals. And in the what we've tested, the S146 is very common in the boar goats, and not exclusively by any means. It's in virtually all the breeds we tried to look, but there were a lot of them, and so we did when we got some animals that were high percentage boar and set up some crosses trying to get the genotype matches that we needed. And so I didn't talk about it, but we in our design, we actually, our homozygous controls, we tried in each case to have 1 or usually 2 that were breed matched as well as genotype matched, trying to go and control for that. And so actually we ended up representing a still small experiment, but a reasonable fraction of germplasm.
I was just curious because as you talked about the European and Britain issue, the boar goats, Savannah goats are coming out of Africa, Kikos are coming out of New Zealand. So, we've got some different, not totally different, especially with the Kiko side because the dairy influence there, but some different, base herd, base populations that some of these are coming out of. So I'm just trying to see if this is going through the way it looks. Just want to make sure that it's out there for all of our breeds. Dairy, meat, everything.
Right.
Fiber.
Sure. So the question is about breed background and what kinds of animals the alleles would be available to and available easily. We have not looked per se at a Kiko, and so that's a good question. We did look. The survey did include Angoras. Angoras on average tended to have a lot less genetic diversity at the prion gene than some of the other breeds. And so I don't think we saw either of these two alleles in Angoras, but virtually all of the other breeds had one or the other. And several of them had, pretty high frequencies, especially of the S. And so in terms of availability, 7 out of 10 breeds, and those included both, meat types like the boar goat, but also dairy types. And so our estimation was that if people wanted to start using that for breeding, that many of them are already going to have this allele and just not know it. And worst case, they'll know somebody who did or something of that sort. Other questions? Thank you very much.
The American Goat Federation, AGF, is a nonprofit national association that represents the interests of more than 200 organizations and thousands of producers engaged in the sustainable production and marketing of goat milk, meat, fiber, and grazing services across the United States. This video was produced as part of the educational services of AGF. AGF also supports research projects that will benefit the goat industry by writing letters to accompany grant requests and providing information when requested. The board of directors is made up of representatives of member organizations, individual producers, and industry specialists from all parts of the United States. Director candidates are nominated and serve 3-year terms on the board. AGF is headquartered in West Lafayette, Indiana, and open from 11 a.m. to 4 p.m. Eastern Time, Monday through Friday. Visit our website, AmericanGoatFederation.org, where you will find information about goat management, healthcare, and other resources. While you're on the website, become a member of AGF and help us provide information to goat owners and represent all goat producers by providing information about our concerns and industry needs to government agencies, USDA, and researchers.